Homeostasis. In biology, homeostasis is the state of steady internal, physical, and chemical conditions maintained by living systems. This is the condition of optimal functioning for the organism and includes many variables, such as body temperature and fluid balance, being kept within certain preset limits. Homeostatic range. Other variables include the pH of extracellular fluid, the concentrations of sodium, potassium and calcium ions, as well as that of the blood sugar level, and these need to be regulated despite changes in the environment, diet, or level of activity. Each of these variables is controlled by one or more regulators or homeostatic mechanisms, which together maintain life. Homeostasis is brought about by a natural resistance to change when already in the optimal conditions, and equilibrium is maintained by many regulatory mechanisms. All homeostatic control mechanisms have at least three interdependent components for the variable being regulated: a receptor, a control center, and an effector. The receptor is the sensing component that monitors and responds to changes in the environment, either external or internal. Receptors include thermoreceptors and mechanoreceptors. Control centers include the respiratory center and the renin-angiotensin system. An effector is the target acted on to bring about the change back to the normal state. At the cellular level, receptors include nuclear receptors that bring about changes in gene expression through upregulation or downregulation, and act in negative feedback mechanisms. An example of this is in the control of bile acids in the liver. Some centers, such as the renin-angiotensin system, control more than one variable. When the receptor senses a stimulus, it reacts by sending action potentials to a control center. The control center sets the maintenance range, the acceptable upper and lower limits, for the particular variable, such as temperature. The control center responds to the signal by determining an appropriate response and sending signals to an effector, which can be one or more muscles, an organ, or a gland. When the signal is received and acted on, negative feedback is provided to the receptor that stops the need for further signaling. The cannabinoid receptor type 1, CB1, located at the presynaptic neuron, is a receptor that can stop stressful neurotransmitter release to the postsynaptic neuron. It is activated by endocannabinoids, ECAs, such as anandamide, N-arachidonylethaноlamide, AEA, and 2-arachidonyl glycerol, 2-AG, via a retrograde signaling process in which these compounds are synthesized by and released from postsynaptic neurons, and travel back to the presynaptic terminal to bind to the CB1 receptor for modulation of neurotransmitter release to obtain homeostasis. The polyunsaturated fatty acids, PUFAs, are lipid derivatives of omega-3, docosahexaenoic acid, DHA, and icosapentaenoic acid, APA, or of omega-6, arachidonic acid, ARA, are synthesized from membrane phospholipids and used as a precursor for endocannabinoids. ECAs mediate significant effects in the fine-tune adjustment of body homeostasis. History. The concept of the regulation of the internal environment was described by French physiologist Claude Bernard in 1849, and the word homeostasis was coined by Walter Bradford Cannon in 1926. In 1932, Joseph Barcroft, a British physiologist, was the first to say that higher brain function required the most stable internal environment. Thus, to Barcroft, homeostasis was not only organized by the brain, homeostasis served the brain. Homeostasis is an almost exclusively biological term, referring to the concepts described by Bernard and Cannon concerning the constancy of the internal environment in which the cells of the body live and survive. The term cybernetics is applied to technological control systems such as thermostats, which function as homeostatic mechanisms, but is often defined much more broadly than the biological term of homeostasis. Homeostasis. An overview. The metabolic processes of all organisms can only take place in very specific physical and chemical environments. The conditions vary with each organism, and with whether the chemical processes take place inside the cell or in the interstitial fluid bathing the cells. The best-known homeostatic mechanisms in humans and other mammals are regulators that keep the composition of the extracellular fluid, or the internal environment, constant, especially with regard to the temperature, pH, osmolality, and the concentrations of sodium, potassium, glucose, carbon dioxide, and oxygen. However, a great many other homeostatic mechanisms, encompassing many aspects of human physiology, control other entities in the body, where the levels of variables are higher or lower than those needed. They are often prefixed with hyper and hypo, respectively, such as hyperthermia and hypothermia, or hypertension and hypotension. Circadian variation in body temperature, ranging from about 37.5 degrees Celsius from 10 a.m. to 6 p.m. and falling to about 36.4 degrees Celsius from 2 a.m. to 6 a.m. If an entity is homeostatically controlled, it does not imply that its value is necessarily absolutely steady in health. Core body temperature is, for instance, regulated by a homeostatic mechanism with temperature sensors in, amongst others, the hypothalamus of the brain. However, the set point of the regulator is regularly reset. For instance, core body temperature in humans varies during the course of the day, i.e., has a circadian rhythm, with the lowest temperatures occurring at night, and the highest in the afternoons. Other normal temperature variations include those related to the menstrual cycle. The temperature regulator's set point is reset during infections to produce a fever. Organisms are capable of adjusting somewhat to varied conditions such as temperature changes or oxygen levels at altitude by a process of acclimatization. Homeostasis does not govern every activity in the body. For instance, the signal, be it via neurons or hormones, from the sensor to the effector is, of necessity, highly variable in order to convey information about the direction and magnitude of the error detected by the sensor. Similarly, the effector's response needs to be highly adjustable to reverse the error. In fact, it should be very nearly in proportion, but in the opposite direction, to the error that is threatening the internal environment. For instance, the arterial blood pressure in mammals is homeostatically controlled and measured by stretch receptors in the walls of the aortic arch and carotid sinuses at beginnings of the internal carotid arteries. The sensors send messages via sensory nerves to the medulla oblongata of the brain indicating whether the blood pressure has fallen or risen, and by how much. The medulla oblongata then distributes messages along motor or efferent nerves belonging to the autonomic nervous system to a wide variety of effector organs, whose activity is consequently changed to reverse the error in the blood pressure. One of the effector organs is the heart, whose rate is stimulated to rise, tachycardia, when the arterial blood pressure falls, or to slow down, bradycardia, when the pressure rises above set point. Thus the heart rate, for which there is no sensor in the body, is not homeostatically controlled, but as one of effector responses to errors in the arterial blood pressure. Another example is the rate of sweating. This is one of the effectors in the homeostatic control of body temperature, and therefore highly variable in rough proportion to the heat load that threatens to destabilize the body's core temperature, for which there is a sensor in the hypothalamus of the brain.